Something is happening right now, inside your body. Something that began before you even put your first bite in your mouth. A cascade of chemical signals, muscle contractions, and molecular decisions are executed with a precision that no human laboratory has been able to fully replicate. The digestive system isn't a passive tube through which food travels. It's a sensory, immunological, and neurological organ all at once. And the most surprising part is that most of its work happens without you being aware of it. Most people think digestion begins when you chew, but science tells a different story. It begins much earlier, when the brain anticipates a meal, when the sense of smell detects the aroma of something to be eaten, or even when one simply thinks about food. The nervous system has already given the order. The salivary glands increase their production. The stomach begins to secrete hydrochloric acid. The pancreas adjusts its enzyme reserves. The body doesn't wait for food. It prepares to receive it with an anticipation that reveals how much of this process is governed by the nervous system and not by the gastrointestinal tract itself. This phenomenon has a name: the cephalic phase of digestion. And it is the first step of a journey that, in terms of total distance, can cover between 9 and 10 meters from the mouth to the anus, depending on the individual. A process that, under normal conditions, can last between 24 and 72 hours. But it's not the time that makes this system extraordinary. It's what happens at each stage. The mouth is the first laboratory. Here, food isn't just mechanically broken down. Saliva, produced in quantities of between 1.5 and 1.5 liters per day, according to standard physiological studies, contains an enzyme called salivary amylase that begins to break down complex carbohydrates into simpler molecules. At the same time, the tongue evaluates the food's texture, temperature, and chemical properties. Taste buds aren't just flavor detectors. They're sending information to the brain, which modifies the intensity of the subsequent digestive responses in real time. A bitter food can trigger protective reflexes. A sweet taste can anticipate the arrival of glucose and prepare the pancreas to release insulin, even before the sugar enters the bloodstream. This is what researchers call the cephalic insulin response, and it suggests that the digestive and endocrine systems communicate in ways that are still being fully understood. When the bolus of food, that moist, chewed mass formed by the tongue, reaches the pharynx, one of the most perfectly coordinated reflexes in the human body occurs: swallowing. In less than a second, the epiglottis closes the passage to the trachea. The pharyngeal muscles contract sequentially, and the bolus is propelled into the esophagus. This reflex involves more than 25 muscles acting in perfect synchrony, coordinated by the brainstem. It is not a simple act. It is a neurological choreography that protects the airway while ensuring the passage of food. When it fails, it has serious consequences. When it works, it is invisible. The esophagus is often the great overlooked part of the digestive system. It is considered a mere conduit, but this muscular structure, approximately 25 centimeters long, performs something remarkable. It does not rely on gravity to move food. Peristaltic contractions, coordinated muscle waves that propagate from top to bottom, can move the bolus even if the body is lying horizontally or upside down. Astronauts in microgravity laugh because peristalsis doesn't need the Earth's pull. The smooth muscle of the esophagus has its own mechanical logic, regulated by the enteric nervous system, a neural network located in the walls of the gastrointestinal tract that some scientists have called, not without some controversy, the second brain. This network contains between 200 and 600 million neurons, depending on the source and counting method, making it the most extensive peripheral nervous system in the body. It operates with a remarkable degree of autonomy. It can coordinate contractions, regulate secretions, and respond to local signals without receiving direct commands from the central brain. Although both systems communicate constantly via the vagus nerve, the majority of that communication flows from the gut to the brain, not the other way around. Approximately 90% of vagus nerve fibers are afferent, meaning they carry information from the gastrointestinal tract to the brain. This fact redefines who is actually in control of the process. Upon reaching the stomach, food encounters a hostile environment by design. The hydrochloric acid present here can reach a pH of between 1 and 2, comparable to the acid in a car battery in terms of corrosiveness. This level of acidity has two primary functions: to destroy most of the microorganisms that arrive with food, and to create the necessary environment for pepsin, a proteolytic enzyme, to begin breaking down proteins. Pepsin cannot function at a neutral pH. It needs this extremely acidic environment, and the stomach, with its specialized mucosa and its constant production of bicarbonate in the protective layer, remains intact against its own acid. When something fails in this protective mechanism, an ulcer develops. The stomach doesn't just process, it also mixes. Through rhythmic muscular contractions, the gastric contents are churned with acid and enzymes until they become a semi-liquid paste called chyme. This process can take between 2 and 4 hours, depending on the food's composition. Fats slow gastric emptying, proteins moderate it, and carbohydrates accelerate it. The pylorus, the sphincter that connects the stomach to the small intestine, regulates the passage of chyme in small portions, ensuring that the next section is not overloaded beyond its processing capacity. And that next section, the small intestine, is where most of what is commonly understood as digestion and absorption occurs. Its dimensions are deceptive. In the living body, it measures approximately 6 to 7 meters in length, but its internal surface area is what is truly extraordinary. The walls of the small intestine are lined with folds called plexi circulares, which are in turn covered with microscopic projections called villi. These villi, in turn, have microvilli on their surface, forming what is known as the brush border. This three-tiered architecture expands the absorptive surface area to between 200 and 300 square meters, according to accepted estimates in the gastrointestinal physiology literature, roughly the area of a tennis court. All of this is compressed within a tube that fits inside the abdominal cavity. But the small intestine doesn't work alone. It is the convergence point for the most complex secrets of the digestive system. The pancreas, located behind the stomach, releases a mixture of enzymes into the duodenum, the first section of the small intestine. This mixture includes lipases for fats, proteases for proteins, and pancreatic amylases for carbohydrates. Simultaneously, the liver, via the gallbladder, releases bile, a fluid that is not an enzyme but performs an essential mechanical function, emulsifying fats, breaking down large lipid droplets into smaller droplets that lipases can efficiently digest. Without bile, fat digestion would be highly inefficient, and without the pancreas, the entire process would be disrupted. This coordination among three distinct organs, the intestine, pancreas, and liver, regulated by hormones such as secretin and cholecystokinin, produced by specialized cells in the intestinal wall, is an example of local endocrine communication that many textbooks present in a simplified way. The molecular reality is significantly more complex, and research continues to reveal additional layers of regulation that were unknown just 2 decades ago. Once nutrients have been broken down into their basic units, amino acids, monosaccharides, fatty acids, vitamins, and minerals, the intestinal wall actively absorbs them into the bloodstream and lymphatic system. Amino acids and sugars pass directly into the portal blood, which carries them to the liver, where they are filtered, processed, and redistributed according to the body's metabolic needs. Fats, on the other hand, cannot enter the circulatory system directly. They are packaged into structures called chylomicrons and transported by the lymphatic vessels of the intestine, the lacteals, until they eventually enter the systemic circulation through the thoracic duct. This detour reflects the chemical incompatibility between lipid molecules in the aqueous plasma of the blood. What is not absorbed, remnants of plant fiber, shed cells from the intestinal wall, water, and indigestible molecular fragments, passes into the large intestine. And here's something that science has taken decades to fully grasp. The large intestine, approximately 1.5 meters long and with a diameter greater than that of the small intestine, harbors the highest concentration of microorganisms in the human body. More than 100 trillion bacteria, fungi, viruses, and archaea live in this environment. They outnumber the body's human cells, even though the total mass of this gut microbiota is only about 200 grams in an average adult. These microorganisms are not tolerated parasites. They are active participants in functions that extend far beyond what was understood just 20 years ago. They ferment dietary fibers and produce short-chain fatty acids such as butyrate, acetate, and prostaglandin, which nourish the colon's own cells. They strengthen the intestinal barrier and have documented anti-inflammatory effects. They produce vitamin K and some B vitamins. They modulate the immune system, which has one of its main training and operational sites in the gut, with more than 70% of the body's lymphoid tissue distributed throughout the gastrointestinal tract. And there is growing evidence, although still in the stages of active research, that the composition of the gut microbiota influences mood, behavior, and possibly predisposition to certain neurological disorders. The proposed mechanism involves the gut-brain axis, the bidirectional communication between the enteric nervous system, the vagus nerve, and the central nervous system. Research in this field has accelerated remarkably since the 2000s, driven by genetic sequencing techniques that allow for the identification of microorganisms without the need for laboratory cultivation. What was once invisible has become mappable. And what this mapping has revealed is that each person has a unique gut microbial composition, shaped by diet, antibiotics, stress, environment, and personal medical history. The gut microbiota of an individual who has received multiple courses of broad-spectrum antibiotics differs significantly from that of someone without such a history. The clinical implications of these differences are still the subject of active research. Water is efficiently reabsorbed in the large intestine. Of the approximately 9 liters of fluid that enter the small intestine each day, from food intake and secretions from digestive glands, less than 200 milliliters end up in the colon. The rest? This fluid recovery function is essential. When it fails due to infection, inflammation, or disease, the result is diarrhea and electrolyte loss that can become dangerous within hours, especially in young children and the elderly. Stool, often perceived as mere waste, is actually a record of everything the digestive system has processed, rejected, and retained. Its composition includes water, shed intestinal cells, live and dead bacteria, mucus, bile salts, and fragments of unfermented fiber. The color, consistency, and frequency of bowel movements are clinical indicators of gastrointestinal health that physicians use as diagnostic clues. Something as seemingly mundane as the yellowish-brown color of feces comes from urobilinogen, a byproduct of the breakdown of bilirubin, which in turn comes from the hemoglobin of aged red blood cells. The digestive tract even processes the recycling of blood components. The final act, defecation, is governed by a system of 2 sphincters. The internal sphincter, made of smooth muscle, operates involuntarily and responds to pressure within the rectum. The external sphincter, made of striated muscle, is under voluntary control. The tension between these 2 systems, one responding to local physiological signals and the other governed by the cerebral cortex, is a small-scale model of the broader tension that runs through the entire digestive system. Local autonomy versus central control. This tension is perhaps the most fascinating feature of the digestive system as a whole. It is a system that can operate, to a large extent, independently. The enteric nervous system can coordinate peristalsis, regulate secretions, respond to local infections, and modulate absorption without direct brain intervention. And yet, emotional states, stress, anxiety, and fear, profoundly alter gastrointestinal function. The gut-brain relationship is bidirectional, constant, and deeply embedded in human biology. What has begun to emerge in the last decade is a picture of the digestive system as more than just a food processor. It is a sensory organ that samples the contents of the tract and sends information to the central nervous system. It is an immune organ that trains the body's defense cells to distinguish self from non-self. It is an ecosystem that co-evolves with the microorganisms that inhabit it and is, on a certain level, a communication system that speaks to the rest of the body, in a language of hormones, nerve signals, and messenger molecules that we are only just beginning to decipher. Disorders of the digestive system are among the most common medical conditions. Irritable bowel syndrome affects a significant proportion of the world's population. Crohn's disease and ulcerative colitis are chronic inflammatory diseases whose incidence has increased in industrialized countries in recent decades, in parallel with changes in diet and gut microbiota. Chronic gastroesophageal reflux, if left untreated, can alter the esophageal tissue in ways known as Barrett's esophagus. Colorectal cancer is one of the most prevalent cancers globally. Understanding the gut microbiota has opened up avenues of research exploring everything from fecal microbiota transplantation to the design of targeted probiotics, with results ranging from promising to still uncertain. The science of digestion is also the science of who we are. Every nutrient absorbed builds tissues, fuels neurons, regulates hormones, and sustains processes from cell division to thought. The digestive system doesn't process food and then abandon it. This process is, quite literally, the material basis of the organism's life. Without it, the rest of the body shuts down, and that's something worth pausing to consider. This system, which works tirelessly throughout life, adapting its rhythm to the seasons, emotional states, diet, age, and each person's medical history, harbors a microbial ecology that weighs, thinks, and responds, communicating the state of the inner world to the brain with greater fidelity than any instrument designed by human technology. This system operates without us ever seeing it, without us thinking about it, without us remembering its existence, except when it fails. That, perhaps, is the most revealing thing of all. The human body has built, over millions of years of evolution, a system so efficient and so sophisticated that it can remain invisible to ourselves. And it is precisely this invisibility that reveals its deepest perfection. What we don't notice is what works best, what's so well designed that it doesn't require our conscious attention to sustain, minute by minute, the life that allows us to think about other things. And now that you see it clearly, everything that happens in your digestive system ceases to be automatic and becomes something profoundly extraordinary. Every signal, every molecule, every microorganism working silently to sustain your life without pause or recognition. The next time you eat, remember that it's not just food that enters your body, it's information, it's regulation, it's biology in action on a scale we're only beginning to understand. And if you want to continue discovering how your body truly works, with the rigor of science and the clarity that almost no one else shows you, subscribe now to Revealed Anatomy, turn on notifications, and join us for the next episode.